chair and the co-chair, the esteemed guest, and my dear students, and of course the organizing committee to give me this opportunity to present here. So my work today is Affimer tagged cubosome, a novel strategy of targeted delivery of drugs to colorectal cancer using recombinant non-antibody proteins. So before I go into my work in depth, let's first look at what are Affimers. So Affimers are basically non-antibody proteins, as the title suggests. So these proteins have been identified or developed using fast display library. So basically these have a very similar to the in structure to the antibodies, like they have a variable region and they have a constant region, and this variable region can be changed depending on what antigen you want to target. So basically these the protein, the corresponding sequence, the DNA sequence is being cloned into a plasmid, and basically as bacterial cells, using a bacterial cells you are able to produce this protein molecule, and these protein molecule have specific affinity to particular antigen, and the antigen which I am working here or presenting today is the carcinoembryonic antigen, which is overexpressed in several kinds of cancer cells, including colorectal cancer. So we clone this the DNA sequence in a plasmid, and then produce the protein molecule, which is the Affimer, in the bacterial cells, that is E. coli cells, and we purified the protein, that is it came around 12 kilodaltons in size, and using surface plasma resonance we further confirm that this particular Affimer has strong affinity towards the CEA antigen, that is the carcinoembryonic antigen. Okay, so this is about the Affimer. So now let's look at what are the cubosomes. So we know that among the several types of nanoparticles which have been researched in the last 20 years, lipidic nanoparticles are the ones which have actually come into the clinic. So they are the most successful nanoparticles. So cubosomes are basically the third generation of the lipidic nanoparticles. So there are three-dimensional structures which are also called liquid crystalline nanoparticles. So they have this aqueous layer, aqueous zone. In between the aqueous zone we have repeated units of this lipidic structure. So in this lipidic structure you can encapsulate any kind of drug, biomolecule, nucleic acid, any kind of fluorescent dye. So these can be easily encapsulated. So what are the advantages of this cubosome over the lipidic nanoparticles? Let's look at that. So these cubosomes have a high encapsulation efficiency. They have way more high stability compared to the liposomes, because they can be stored at room temperature to up to 6 weeks, whereas the liposomal formulations are generally stored at cold temperature. They can be easily conjugated with proteins, antibodies, or any targeting ligands, because these cubosomes can be easily functionalized with different kind of functional groups. For this particular work, I added azide group for easy the click chemistry coupling. And these cubosomes can easily evade the mononuclear phagocyte, so that is why they can retain in the circulation to up to 72 hours. And there are several reports which claim that these cubosomes can also penetrate the blood-brain barrier. So they can cross the blood-brain barrier, and thus the drugs can be easily delivered to the brains. Okay, so now let's look into my work. So this is the cubosome which we synthesized, and in the cubosome we encapsulated a copper drug, which was a organometallic complex. So this copper drug did show some cytotoxic activity in cancer cells, but it was not like specific. So it was also killing the non-cancer cells as well. So we encapsulated the drug in the cubosome, and we functionalized the Affimer proteins, the proteins which can target the carcinoembryonic antigen, on the surface of the cubosome. So this is the HR-TEM of the cubosome nanoparticle. As you can see, the name suggests it's cubic in structure. So you can easily see the structure is exactly cubic. And this is the cryo-TEM image of the same cubosome. So you can easily see the internal structures in which the drugs actually get encapsulated. Okay? And using the single X-ray scattering data, we analyze the internal structures. So I'm not going into that characterization detail. So we finally got 142 nanometer size cubosomes, which was encapsulated with a drug, and the Affimer was conjugated. Now, as I said, the antigen which we are interested, or we are trying to target, is the carcinoembryonic antigen, which is well reported to be overexpressed in various cancers, including the colorectal cancer cell line LS1740. And this antigen is not expressed in any normal cells. So HEC 293 is one of the negative control for our study. So using fluorescence study, we found that this CEA antigen is overexpressed in the colorectal cancer cell line. The green fluorescence is coming from the antibody staining, whereas HEC does not express, and same was further validated using Western blot. Then we wanted to say whether this Affimer can actually drive the cubosome to cancer cell or not. So what we did was we encapsulated a green fluorescent dye in the cubosome, and then we used the same cell line and we tested in two different groups. In one group, the cells were treated with the Affimer conjugated cubosome, and another group there was no Affimer but the cubosome with the dye. As you can see, in the Affimer targeted group, there is specific uptake of the cubosome showing the green fluorescence in cytoplasmic region, whereas the non-targeted did not take any uptake. So this indicates that the Affimer conjugated cubosome can actually go in the cancer cell. So next we encapsulated drug in the cubosome, and we found that in the same cell line, that is the colorectal cancer cell, when you are targeting, the red bars are the targeted group, there is specific significant cytotoxicity observed, but in case of the non-targeted, where there is no Affimer, the drug encapsulated cubosome is not causing any cytotoxicity. The same is also found in the same panel with the HEC cell lines also. So if you look at the HEC cell line particularly, you can see that in case of the targeted delivery or non-targeted delivery, there is no cytotoxicity observed, but only cytotoxicity observed in case of the Affimer targeted group in the colorectal cancer cell line. Okay. So next we wanted to further analyze, before going into in vivo model, we wanted to analyze in using the 3D spheroid model. So we developed 3D spheroids of both cancer cell line and the HEC293, which is a non-cancer model. And as you can see from the red fluorescent data, that there is specific cytotoxicity observed after Affimer targeting in the 3D spheroid model, but no toxicity at all in the non-cancer cell line. And the same finding was concluded using apoptosis, using caspase-3 marker. So in the LS1740, after Affimer targeting, there is significantly high amount of apoptosis observed, but none in case of the rest of the groups. Now it was time to check our efficacy in vivo. So we developed xenograft model in nude mice, and using the same cell line, LS1740, which is a human cancer cell line. So in xenograft model, we wanted to check whether the Affimer can drive the cubosome to the tumor region or not. So we encapsulated Cy5 dye in the cubosome, very similar to my in vitro work where we encapsulated a green fluorescent dye. And after Affimer targeted, you can see these are all the organs like heart, liver, kidney, spleen, brain, lungs, and tumor. So this middle one, which is number 4 here, is the tumor. And after targeted delivery, you can see there is specifically high uptake of the Cy5 fluorescence cubosomes in the tumor, and very negligible amount in some liver and spleen, which is quite commonly observed. But in case of non-targeted, there is no uptake in the tumor region. So this concludes that the Affimer conjugated cubosome can be specifically driven towards the tumor in the mice. Okay, and this is the quantitative data analysis. And the same was further concluded from the tissue sections. So the same tissues were cut into microtome, and we finally observed that in targeted group, you can see the maximum fluorescence in the tumor region, but in non-targeted, there is none uptake. Okay. Now it was time to check the efficacy of drug after delivery in the tumor xenograft. So we developed the same tumor xenograft model in the mice, and using ultrasound imaging, we measured the volume of the tumors. So normally people use caliper, but the USG gives a more precise tumor volume measurement. So we used the USG measurement, and as you can see in the Affimer targeted group, the tumor size is quite restricted, whereas in the non-targeted or control group, the tumor is quite big. So this is kind of indicating that after drug delivery of the Affimer targeted cubosome, we can restrict the tumor growth. And further, it was also the quantitative data is here. So you can see in the Affimer targeted group, the tumor growth is completely restricted compared to the non-targeted or control group. This is the fold change increase in between the days of the treatment. And further, we want to see whether this observation is actually coming from drug release or not. So because my drug was copper-based drug, so we did LCMS, and we analyzed the drug uptake in various tissues. So we found in the Affimer targeted group, there was maximum drug uptake in the tumor and very negligible amount in the rest of the organs. So further we want to see whether the tumor shrinkage was actually due to the cell death in the tumor or whether it was some other effect. So we further took the tumor tissues of the three groups and analyzed them using H&E staining, and we found that in the targeted group there was significantly high amount of necrosis, which is obviously after the apoptotic event we see a necrotic tissues. So the high amount of necrosis was found in the targeted group, very less in the non-targeted, and of course nothing in the control group. So this is the quantitative data of this representation. So the next, the final thing which needs to be seen is whether this targeted delivery is causing any cytotoxicity in the rest of the organs or not. So we took all the organs of the mice, all the important organs of course, the kidney, liver, heart, lung, brain, spleen, and we did analysis of the tissue sections on the three groups, and we did not found any cytotoxicity in any of the tissues. So this was my work which we published in the year 2023, sorry 22, and if you are interested, you please feel free to go through it. And I'm very open to collaborations currently. So if you are interested in using these cubosomes or the Affimer protein, so as we were discussing earlier that antibodies are very costly, right? So Affimer proteins are a good alternative for these antibodies because they are produced in the bacterial cell. They don't have any immunogenic reaction in the mice body. That shows that they are pretty safe to be used, and they have quite promising effect, very similar to antibodies, but we are cutting the cost because these are produced in bacterial cells in a bulk amount. Okay, so just using a bacterial cell, you can just produce the protein and you can purify it, and then you can use it for tagging in cubosome kind of nanomaterials, or any other nanomaterials are also possible to be conjugated. So that's all for my work. Thank you. If you have any